Hello students, in today's class we are going to learn about linker and adapter molecules which are used in cloning reaction, and we will also learn about topoisomerase based DA cloning method. So linker molecules are basically tecomeric molecules which contain an internal restriction endonuclease target site. So here we have a eco R1 linker molecules containing 10 base pair sequence containing an internal eco-R1 target site. So during the cloning reaction, we have our foreign DNA molecule that is to be cloned, and we add the linker molecules first to either end of the foreign DNA molecule. For ligating them to either end of the foreign DNA molecule, we mix the two in presence of enzyme DNA ligase, and that will add linkers on either side of your DNA molecule to be cloned. We then digest with the restriction endonuclease. Similarly, we need to digest a vector molecule as well with the same restriction endonuclease. We then mix the two, the vector and the insert, in presence of enzyme DNA ligase, and ligation reaction will lead to generation of a recombinant vector containing the insert molecule. Then we need to do transformation and a screening step in order to identify the clone containing the product or the required insert. Next, we have adapter molecules. So adapter molecules are molecules which contain preformed cohesive ends. Okay, so in the previous example, that is linkers, we have seen that they don't have preformed cohesive ends. They basically have blunt ends, and they can help with the cloning reaction by ligating them to either end of the insert molecule. But sometimes, like in case of linker molecules, after ligating to the insert molecules, we do a restriction digestion step. But sometimes we cannot do the restriction digestion later because of the presence of internal restriction enzyme target site present within the insert molecule. So that is going to fragment your insert molecules. So instead we can make use of adapter molecules. So adapter molecules are already digested with restriction enzymes, and they contain the hydroxyl group and phosphate group present at one end, and at the other end they contain hydroxyl group present at both the sides, and this is going to prevent the self-ligation molecules of the adapters. Okay. So now we mix the insert molecule with the adapters, and adapters will be ligated at both ends of the insert molecule, which are having preformed sticky ends. Then we need to do a use of a enzyme polynucleotide kinase that will add phosphate group at the 5' end, and now we will have phosphate at the 5' and hydroxyl group at the 3' end. Similarly, we need a vector molecule which is also having the same type of sticky ends as contained in the adapter. So mixing of the vector and the insert molecule in presence of enzyme DNA ligase and the ligase buffer will allow the ligation reaction. Now the third point that we will discuss is about topocloning method, which is also known as TA cloning method, that is based on the use of the enzyme DNA topoisomerase for cloning purposes. Now, I hope we remember the role of DNA topoisomerase. It is basically involved in DNA replication process. So during DNA replication process, because of the activity of enzyme helicase, there is unwinding of a DNA, and this leads to generation of the torsional stress in the rest of the DNA. And this torsional stress can be relieved or removed with the help of the enzyme DNA topoisomerase. So what DNA topoisomerase does is it can bind to both strands of DNA. However, DNA topoisomerase 1 acts on only one strand, that is, it cleaves one strand of DNA. So this leads to the breaks, and the other strand will pass through the break and will decrease one number of supercoil. And then the topoisomerase will reform the phosphodiester bond in the backbone, and your number of supercoils in DNA will decrease. Okay. So normally when we do a cloning reaction, we have to make use of restriction digestion and then a ligation reaction. But here, by looking at this activity of the enzyme, people propose to use TA cloning method based on the use of enzyme topoisomerase, wherein we can make use of two different topoisomerase enzymes to cleave the two strands of the parental DNA. Okay, because topoisomerase 1 cleaves only one strand of the parental DNA, and we need cleavage of both strands of the parental DNA, just like digestion with restriction endonuclease, so that the insert molecule can be ligated. Now the next slide explains about the mechanism of topoisomerase cleavage. And here we can see the initially supercoiled, you can say, form of DNA, which is having 4 supercoils. Now, so here we will have the activity or binding of enzyme topoisomerase. So topoisomerase molecule basically contains a tyrosine residue within its active site. This is the structure of amino acid tyrosine. And within its R group, it contains a hydroxyl group. So this hydroxyl group is basically going to act as a nucleophile, and it attacks the phosphate of the phosphodiester backbone. And in this process, the hydroxyl group of the tyrosine residue will form a bond with the phosphate group, and the other end will contain a hydroxyl group now. So energy is basically stored here between the new bond that is formed between the phosphate and the tyrosine residue. Then after that, we will have passing of one strand of the DNA through the break. And after completion of the process, the free 3' hydroxyl end will now act as a nucleophile and will basically displace the tyrosine molecule. And in the process, it will itself form or reform the bond between with the phosphate of the backbone, and in the process it will displace or it will remove the enzyme topoisomerase, and the energy released during this process is reformed or is locked up while formation of the previously formed phosphodiester bond in the backbone. So this is the basic mechanism how by which the enzyme topoisomerase works. And as you can see at the end of the process, the number of supercoils will decrease to 3 from 4 supercoils. So these same points are written here for your reference. So now we will discuss about the mechanism of TA cloning. So this enzyme, we basically need to make use of a specialized vector for this cloning reaction, which is known as the topocloning vector. It contains various genes such as the kanamycin and the ampicillin resistant gene for the selection of the vector, and it contains a puck origin of replication. It also contains a LAGZ gene, and upstream of which is present a promoter for the LAGZ gene. And towards the start of the LAGZ gene, we also have a multiple cloning site. So here, since we are not doing restriction digestion, the multiple cloning site is not relevant. But what is important is actually a specific signal sequence, which is recognized by enzyme topoisomerase, which basically consists of these thymine residues present on both the strands of DNA, and they have specific 33 base pair sequence both downstream and upstream of these thymine residues, which are then also flanked by these sequences in repeat sequences in yellow. Now, topoisomerase will specifically bind to these thymine residues and cleave the DNA. So, after digestion or after the activity of topoisomerase, you will basically generate these thymine overhangs on the vectors, and the topoisomerase enzyme will actually be present bonded to the thymine residue. Then simultaneously we need to prepare our insert molecule. So insert molecule is prepared by PCR reaction by making use of Taq polymerase enzyme for the amplification process. So Taq polymerase enzyme basically has a property that automatically towards the 3' end it will add one adenine residue during extension, and the addition of this adenine residue is in a non-template based fashion, and this will lead to generation of a terminal adenine residue in both strands or a terminal adenine overhang. Now you can observe that actually these adenines and the thymine residues are complementary with each other. So in the next step, what we basically do is we add the vector, then we add topoisomerase enzyme and Taq amplified PCR product, and we mix and incubate the reaction for about 5 minutes at room temperature, and automatically the ligation reaction will be completed. As you can see over here, it will automatically the fragments will be ligated. The mechanism for ligation here is the hydroxyl group. As we discussed previously, the hydroxyl group that are present at the ends, they are going to make the second nucleophilic attack that is going to displace the topoisomerase, and in the process it will form the bond with the phosphate of the thymine residue. So in this process we will have bond formation, and at the end we will have intact DNA or the ligated DNA with the vector. The next step is basically transformation and screening step in order to identify the correct flow. So the mechanism for TA cloning is, the important points are written for your reference. And coming to the importance of the TA cloning method. So if we are doing a normal cloning reaction, we need to do restriction digestion of the plasmid vector. We need to do digestion of we need to do first the PCR amplification, then digestion of the insert. Then there is a cleaning step where we column purify both fragments. Then we do a ligation reaction. Ligation reaction is generally and 4 to 5 hours minimum or overnight reaction. So overall, we can say that overall the restriction digestion and the ligation steps may take about 1 to 2 days for completion. While by making use of the TA cloning method or the topo-based cloning method, we can actually complete the entire experiment in about 1 to 2 hours, and after 1 to 2 hours we can enter into the transformation step. So we can it is basically a time saving method, and it is less tedious because we can combine uh, the different steps into one step, that is digestion and ligation can be combined. So that's it about adapters, linkers, and TA cloning method. Thank you very much, and kindly like, share, and subscribe if you like the content.